Okay, I'm going to start. It's my favorite topic. CI, this is what I study. I can actually use my own papers. Okay, so this is basic CI. It has to do with the infection status of the insect. And this is with Wolbachia, remember. So Wolbachia is a bacteria that lives inside of the insects, lives in the gonads, in the ovaries, and it causes this reproductive phenotype called cytoplasmic incompatibility, which is essentially sterility that can be rescued like a toxin-antidote system. So in males, if you just draw out the Punnet Square, if you're a male and you have Wolbachia and you're infected, okay, and you mate in this box with a female that's uninfected, all these offspring die. And that's because the Wolbachia that live in the male, they do some kind of a modification that causes a sperm to malfunction, and that female that doesn't have Wolbachia can't rescue that, so they all die. Okay, toxin-antidote system, the flip side of this is that the female, if she's infected, if the female's infected with Wolbachia, those females produce an antidote in the eggs, and that female can combine, or if she mates with the infected male with the messed up sperm, whatever the rescue factor is, it can fix the sperm, and these all survive. So if you're female, if you mate with an uninfected male here, or here with an infected male, all your offspring survive. If you are not infected, this one, and you mate with an uninfected male, you're fine. But if you accidentally mate with a male that is infected, all your offspring die. So this female is at an advantage. Because it doesn't matter who she mates with. All her offspring always survive. And all her offspring always inherit Wolbachia. So it perpetuates the system and spreads Wolbachia like we talked about yesterday. This is what I did my PhD on. And this was like in, let's see, I started my PhD in 2009. In 2009, nobody knew what the genes were in the bacteria that caused the CI. We just kind of knew that it happened, and nobody knew how it did what it did. Okay, so our first experiments were, well, we made a couple assumptions. One, we knew that CI was causing some kind of a modification in the sperm. We knew that was a fact from the previous literature. And we knew that as sperm develop, so you have a germline, we talked about this in development. There's a germline, which is like stem cells, okay? And then that germline will divide. The niche is right here. This is where these things stay as stem cells, but then as they divide, each time you get more, okay? This is in the testes, and all these cells are linked with what are called ring canals, okay? And eventually, they become sperm, right? So if this is a testes, this is the germline niche where the stem cells sit, and they divide, and then there's morphogens along this pathway that tell the sperm to differentiate into adult sperm. And we knew about Wolbachia that Wolbachia infect the germline, and they infect some cell stages in spermatogenesis up to, these are basically, you don't have to remember this, but these are spermatocytes. Spermatocytes. This is the phase where they're not quite sperm yet, but they're like becoming sperm. And Wolbachia infect these. But when the sperm become adult sperms, what they do is they basically have to make their cell become kind of like aerodynamic. It needs to be able to swim through viscous solutions. So the whole morphology of the cell basically adapts and changes to become like a swimming machine. And that's why you get the tail, and in the tail are like little motor proteins that cause the thing to go like this, like you've seen. And when this happens, the cytoplasm has to get removed. Cytoplasm must be removed, because if you think about it, cells are like balls and sperm are basically like that. So to get from here to here, you have to actually like remove the cytoplasm. So there's a process that happens from this transition here to here where the cell basically gets squeezed and it elongates, and it squeezes out all the cytoplasm out the back end. So if you can imagine just like pressure from here and here, squeezing it together, and then you move that pressure all the way down, just like a toothpaste tube, all the way down to the back, you squeeze it all out. and then it gets removed out at the end in what are called waste bags. Okay, and Wolbachia also gets squeezed out. So the Wolbachia gets squeezed out. So this means, what does this mean? This means that this is two, that Wolbachia are not present in adult sperm. So then let me ask a question. So then how can sperm cause CI? How can sperm be modified if there's no Wolbachia there?
There's a big toxins in the sperm.
Yeah, like it leaves something behind. That was our conclusion, that whatever CI is, whatever it is, it's going to leave something behind. Okay? The Wolbachia is going to leave something there, factor X, that's going to do something to the sperm. Okay? And we thought we should be able to detect this, whatever this is. We thought it was a protein. If it's a protein, we thought we should be able to detect it by liquid chromatography, mass spectrometry, and maybe SDS-PAGE. And the reason we thought that is because if you take males that have Wolbachia in an insect, and you mate them repeatedly, so you mate them to female 1, then to female 2, then to female 3, then to female 4, you get high CI here, but weaker CI here, weaker CI here, weaker CI here. That, when I say weak CI, that means like there's less killing of the embryos, less kill rate. So let's say the CI here is 100% kill rate. Maybe you'd see like 99% in the second mating to the second female. Maybe you'd see like 95%. Maybe you'd see like 90% killing. What does that mean? What do you conclude from that? From me telling you about these experiments, what can you conclude from that?
Whatever's calling it is decreasing over time.
Yes, and that's actually a significant thing, because that means there's a limiting factor, and that means that to get CI, you need like a bunch of it. You know what I'm saying? You need like a lot of it. You got to like stockpile it, because those stockpiles kind of like run out if you do these mating experiments where you keep mating. So we thought, well, that kind of suggests that if you did mass spectrometry on the sperm, you should kind of be able to detect those stockpiles of whatever the thing is. You should be able to detect the stockpile of that toxin or that protein. That was our assumption. That was my assumption when I started my PhD. Okay, so then we thought, well, then all you need to do is you need to take a female that doesn't have Wolbachia. Okay, so you take a female mosquito, and she's got an organ, which is like 3 little balls in her abdomen that are called spermathecae. What do you think they do?
Store the sperm.
They store the sperm. Did I already tell this story? No.
They fertilize the nest and play them.
Okay. So she's got organs that store the sperm. All you need to do experimentally to figure out what the factor is you need to take an infected male, do this cross, so you mate them, and then you take that female. Now she's got these spermathecae that are filled with sperm from the male, okay? And then you can dissect out the spermathecae. So you can actually dissect insects. You could dissect bugs, take a needle, you like tear it apart, and then these balls will like fall out, and then you collect the balls in like a tube. So I was like collecting mosquito spermathecae for forever, dropping them into like this little tube, and they're all filled with sperm, and they're all filled with sperm that came from the infected male. Okay. So basically what I'm telling you is I can collect sperm from infected males. And here, when I draw red, it doesn't mean it's infected with Wolbachia. It just means it came from an infected male. And then I did the same experiment, except instead of infected male, I used uninfected male. Right? So he mates, and then I collect the uninfected spermathecal balls. So I needed like, it was something like 800 of these. So it took me actually like 2 weeks, 40-hour work weeks to collect these spermathecae to get enough for an experiment. It was horrible. But when you get to the point where you have these, you can do a plus minus comparison, right? This is basically infected sperm versus uninfected sperm. It's basically the gist, okay? And then I would, 1, extract protein, okay? And then I would, 2, run an SDS-PAGE with some modifications. And I would basically just like compare the proteins. So let's look at that data. That's in this paper. Is it too dark? Let's zoom in on the figure. Can you see these? Can you see that? That this is different from here. There's something here that's not over there. So this is SDS-PAGE of all the proteins that appear in sperm samples from lane 1 is infected males. Lane 2 is, I can't change my color here, but lane 2 is uninfected males. So here you're just looking what's present here but not here, and you see these 2 bands, right? And we also did this in females, in the female ovaries. So we dissected ovaries, and we found this, and oh my gosh, it's going haywire, and that, okay? And we saw, okay, so then what we did is, so we were the first people to detect, like there's a visual difference. There's at least 1 to 2 proteins that are present in the sperm, but not present in uninfected sperm. And we knew the rough size, which was about 56 kilodaltons. Okay, so then we can cut out these bands with a razor. And we do mass spectrometry. This isn't a test on, or this isn't a class on methods per se. So I won't talk too much about that, but I'll just say it's a way to tell you what proteins are there, what proteins are present. And the way that it can do that is it can detect a mass, and from that mass, it can determine a little sequence of peptides that's likely. So let's say you have a sequence of peptides K, A, M, Q, G. And then you can take that sequence and you can basically blast it against a sequence genome to figure out where that peptide came from. That's basically mass spectrometry in a nutshell. So you can get a list of all the proteins that are present in that little gel slice, okay? So we did that, and we detected a Wolbachia protein that was present, was constituting that band, okay? So we didn't have a name for it at the time. We just called it its name in the genome, which was WPA0282. That was when you sequence genomes and you don't know what the proteins do, you name them numbers. And so this was the protein we found. But this was a significant advance because we were the first people to show that there was at least 1 protein from Wolbachia that was being stockpiled in the sperm. And we knew it wasn't like a structural protein of Wolbachia, like the surface protein or something like that, because there are no actual Wolbachia in the sperm. So we knew it was being secreted, pumped out, stockpiled in the sperm. Okay. So then we looked at where they were in the genomes. So look at this figure. So here you see 282. So this is the protein that we detected. And there's different strains of Wolbachia. Okay, there's a strain from Wolbachia that lives in mosquitoes. There's a strain from Wolbachia that lives in Melanogaster, Drosophila, that's fruit flies. There's a strain of Wolbachia that lives in Drosophila simulans, that's another fruit fly. So one of the things is these all cause CI, these different strains of Wolbachia. They all cause CI. So if this thing that we found stockpiled in the sperm was somehow related to CI, you would expect to find that gene in all the CI-causing Wolbachia genomes, right? Does that make sense? So all we had to do was blast the sequence of the gene to check that it was in all the CI-inducing genomes, and we did. We found it. So here's 282. Here's its ortholog in Drosophila. Here's its ortholog in Riverside, Wolbachia from Drosophila simulans Riverside, and there were actually multiple copies of it. Okay, and the first thing that we noted was that it was part of a 2-gene operon. It looked like this. So you see these 2 black arrows, that was basically a synthetic operon. What's the synthetic mean? I know I've defined that before for you guys. Yeah, it's conserved order. So look at the order in these three different strains. This one, and this one. It's always 282, then 28. Well, the numbers are different, but it's always this one and then this one, always in the same order. It's never flipped. It's never broken. It's never like pieced apart. They're always together and they're always in that order. Okay. So we knew it was a synthetic, it was organized as a synthetic operon. And one way to prove that it's an operon is what are the characteristics of operons that we've talked about before?
It's conserved.
Yep, we got that. We checked that. One promoter. That's more difficult to test. We did not test that. We got criticized for that. Also, polycistronic transcription. Remember, what was that?
The RNA reads all the way through. There's no stop.
Yeah, the RNA polymerase pops on right here, and it makes one transcript. So you can check that. And the way that you check that is you design a primer here and a primer here, and you do a RT-PCR, which is reverse transcriptase, which will PCR the transcript. So your primers will go to the transcript. And you can amplify, if you can amplify this region, which corresponds to this intergenic sequence, then you've proved that that is a polycistronically transcribed operon. So I'll show you that data. So we did that right here. So here's 282, here's 28283. I'm drawing the picture for you exactly what we're doing. We're checking to see, okay, are these genes polycistronically transcribed? Are these genes polycistronically transcribed? Are these ones polycistronically transcribed? Okay, and what you want to look at here is this box is this reaction, this box is this reaction, and this box is this reaction. And the DNA, so here we're doing, this is a positive control. This D, that stands for DNA. We're doing a positive control. That means we're checking to see if our primers work. So it's always going to work on the DNA. That's where, so here I said this is the transcript, messenger RNA. Here's the DNA. You're always going to be able to PCR across the intergenic gap in the DNA, right? Does that make sense? Okay, but you're not necessarily going to be able to PCR across the gap in messenger RNA if it looks like this. If there's two transcripts, you won't be able to link those primers. Does that make sense? So it's going to work to PCR the DNA, but if it's not an operon, it's not going to connect to two completely separate transcripts. Does that make sense? Or am I going too fast? Okay, so here, so we tested the DNA, there's a positive control, and we tested in the females and the males. And what you're looking at are these, and there's no bands. That means it's not being transcribed across this piece. Now here is the positive control DNA, and here is the band. Here is the band you want to look at. It's this in the males and the females, it's being transcribed across the gap. So you see that band, that's what that means. What is this control, minus RT?
No reverse transcriptase.
Right, no reverse transcriptase. You would expect, if you're actually PCRing the transcript and you don't add reverse transcriptase, you should not get a band there. Does that make sense? So that's a negative control to make sure that whatever you're getting is not a product of DNA, it's a product of RNA. Okay, so you see here, it's a perfect control where you have a positive control, a negative control, and then there's the band. And we could cut out that band and sequence it, and it did correspond to this intergenic gap right there. And then here we do the same thing with this piece, and there's no bands. So it shows you that it's not being transcribed across this space or this space, but it is here. So this is the operon. So we define that as an operon. This is the same thing, but with a different paralog. So paralog is when you have a duplicate of the same operon. We checked it with the paralog, and it worked even better in the paralog. The data is even nicer. That's not worth talking about. That's a control. Okay, so we proved that this thing that we found, 282, this protein that we found in sperm, it's part of a 2-gene operon. Why is that important? Why would we care about that? Like, why do we care that it's 2-gene operon?
Are both of those proteins found in the sperm?
That's unclear. Nobody has detected the second one, but nobody's looked for it. So when we did the gel, I get criticism for that all the time. When we did this mass spectrometry experiment, we only cut out these bands. That second protein is a lot bigger and it would have been up here. So you can't really see it, but we never cut anything out. What I really wanted to tell you is, like, why is it important that it's an operon? The reason it's important is because when you look at plasmids in bacteria, what do toxin antidote systems look like? They're operons. They're organized as operons, and they're always organized in the same way. You always see the antidote and the toxin in this order, 5' to 3'. Okay? You see this all the time. This is a common pattern in microbiology. These toxin antidote systems are operons. And we knew that Wolbachia, okay, remember I just told you that Wolbachia acts like phenotypically like a toxin antidote system, right? We knew that it looked like a toxin antidote system, but nobody had ever found the genes. So to find a gene that's in the sperm that is organized in something that is consistent with a toxin antidote operon suggests that maybe this one is the antidote and this one is the toxin, right?
But you found 2A2
this one in the sperm. Yes, we get that. You guys are smart. We found that one in the sperm. And so what I told you is point one. You can say, some people say, well, you didn't find that one in the sperm. That's the one you really need. Okay, the point to that is that we weren't looking. We never actually measured that. We weren't looking. The second point is, remember how toxin antidote systems work, right? I've drawn this out. So here's the bacteria that's dividing. Here's the plasmid. Does it make just the toxin or does it make both? It makes both. So here's the antidote. Here's the toxin. And then what happens? What happens to the antidote?
Whenever it splits, the antidote degrades first.
Right, the antidote degrades. So now imagine how similar this is to the Wolbachia system. Let me just draw it out. Okay, so then it would die after the thing degrades. Although it would, if you don't have the plasmid, then you would die because the antidote degrades. Now imagine the sperm. Okay, we got a figure for this. This is in the review that I assigned. Here's the figure for that. Okay, so here's the spermatocyte becoming a sperm. And look what's happening. The Wolbachia are making both. The green thing is the antidote. The yellow thing is the toxin. Okay? So it's pumping out both of these things in the sperm. And our hypothesis is that if this sperm combines with an egg that's uninfected, this is the same scenario as a bacteria not inheriting that plasmid. It's not inheriting the Wolbachia. So is it going to be producing more antidote? No, because there's no Wolbachia there. So there's no more antidote, and then our hypothesis is that the antidote degrades first, because that's how all toxin antidote systems work, okay? And then all that's left behind is the toxin, and then these embryos die. Now here's how a rescue is postulated to happen. So if this is the infected egg, okay? So you got that sperm, it's got both toxin and antidote in there. It combines with the egg, but the egg has Wolbachia in there, which are these little red candy-looking things. And they're perpetually making more antidote, right? In higher stoichiometries to the toxin. So this leftover antidotes can pick up these toxins and basically save the system, and nothing happens, and so these embryos survive. So that's the hypothesis. Does that make sense? So that's how we think it works. So yes, we didn't per se detect this, but we should expect to see both antidote and toxin in the sperm. And which one would we actually expect to be at higher levels? The antidote. Right. And it's every time it's made, it makes more antidote. In toxin antidote systems, they always make more antidote than toxin. That's usually how they work. So we would actually expect to see more antidote in the sperm. So yes. But the point is, the basic point is, like, you can disagree with some of the little parts of the hypothesis at that point, although we came and proved it later. At this point, basically the important things were, one, we were the first people to detect a Wolbachia protein in the sperm. So either way, you might start to think, well, that thing might be involved in CI. And we proved that it was organized as a synthetic 2-gene operon. And then we postulated that this probably is like a toxin-antidote operon. Okay, so that was the first paper on CI during my PhD. Then we published the next paper. Okay, so then we got our Nature Microbiology paper. This was a monumental effort. Okay. So here we just diagram out what CI is again. Here we're diagramming out some different orthologs or homologs of these different systems in different Wolbachia, different paralogs. So the first thing is this figure. Okay, so let's go to the notes. So in a toxin-antidote system, how does rescue happen? Even in a bacteria, how does the rescue happen in a bacteria? At a molecular level, what's happening?
The antidote like binds to the toxin so it can act.
Yeah, this is the 90%. These are called protein-protein TA systems. In protein-protein TA systems, that means the antidote is a protein, the toxin is a protein. They bind each other, okay? And the antidote will inhibit the toxicity somehow by basically just binding to the toxin. So there's a binding interaction, okay? We can test for binding with a pulldown. What's a pulldown? What's a pulldown? Where have you heard of a pulldown? Don't let me down.
I think you run it over some beads like that.
Okay, what would bind the beads?
Like a histidine tag or a line.
Thank you. Okay, so if you put a hip epitope tag, like a His6, in front of, let's say, your toxin, and you grow it in E. coli, I can make a plasmid. I did it with the ERA operon. Okay, so it's got the ERA operon. I put the gene, the actual name of this gene, the toxin, the second gene, we call that SID-B. But you can just think of it as His6 toxin. So I built that plasmid. You can express it in E. coli. What would you add to induce the toxin? You add arabinose. Okay, and then you grow these bacteria, you lyse them, you run them over the column. If these are cobalt or nickel beads, they're going to grab the toxin, and all these beads are going to be bound to His6 toxin. Does that make sense? Okay, that's the first part of the pull down. That's the bait. If you read papers, you'll see this as bait. Okay, so I have column set up that's completely bound to His6 toxin. Okay, then I can make a second plasmid with flag, it's a tag, antidote. And I'll put it in front of the arabinose operon, clone it in here. And I can grow this in a separate line of E. coli. I can induce with arabinose, and that's going to make flag antidote. Okay, then I take this bacteria that I lysed, I took the lysate, and then I put that lysate on top of the column. So this is step 2. This is the second thing. This is the pull down. This is the prey, right? This is the thing that my bait is trying to grab. Okay, and if there's an interaction between this thing and the antidote, is the antidote going to drip through the column? It's going to stay, right? So it's going to stay if there's an interaction. If there's no interaction, it should just drip off the column, right? Does that make sense, Avery? Okay. So we do this, and then you wash a whole bunch of times. You just throw buffer on 50x column volumes of basically wash buffer, which causes everything to drip out. And the only thing that's left behind is whatever is binding to the toxin. And if the antidote is binding to the toxin, then I can step 3. Step 3 is elution, elute, which means I add in something that has a higher affinity to those cobalt beads like imidazole. And then these 2 proteins are going to fall off, and I can collect them in a tube. Okay. And then I can run an SDS-PAGE or a Western, which is what I did, on this sample to see what my toxin was binding to. Does that make sense? Okay. So that's the setup for this data. So that's this data. Okay. So here what I'm telling you is I got the His6 toxins. Oh, I can't erase. This is a negative control. What is this protein? Yeah, it's LAC-Z. That's the beta-galactosidase. That's the enzyme that degrades lactose. Why is it a negative control?
It shouldn't bind to the antidote.
Right. There should be no interaction with some random protein, LAC-Z, and the antidote. So this is a negative control where I do the same experiment with LAC-Z. And here what I'm doing, this is a Western blot. So this is anti-flag. What does that mean? Right, the antibody is detecting the flag. And here I have 3 different types of antidotes, all flag tagged, okay? And what I did is I mixed each toxin with 3 different antidotes, okay? So I have these 3 operons, these are paralogs. Remember back when you asked this question about the one Wolbachia strain to a different Wolbachia strain when you get CI, and I said that's called bidirectional incompatibility, and I said the reason for that is because that toxin antidote system doesn't match that toxin antidote system. That's because there needs to be a matching binding between the antidote and the toxin, right? We can actually test that hypothesis by this experiment. So I grabbed different toxins from different strains of Wolbachia that are orthologs or paralogs, but not the exact same gene, okay? So I have 3 different antidotes, 3 different toxins, 2 different toxins, okay? And here I mixed this one, His6 SID-B, that's a black version that matches the black one. Okay, and I mix it with these 3 different antidotes. And I'm telling you, this is the one it actually matches. Okay, and these ones it doesn't match. Is there any interaction with the ones that it doesn't match? No interaction. There's only an interaction with the one it matches. And the reason you're seeing it is because this has a flag antibody, or a flag epitope tag, and I'm doing a flag Western, which will bind these sequences and light them up. Okay. Now, if I take the different toxin, the red toxin, okay, and I mix the red toxin with the red antidote, there's an interaction, but there's no interaction with its non-cognate matching partners. Does that make sense? So what I proved here is this is a pull-down experiment that proves 2 things. One, it proves there is an interaction between the first gene and the second gene. 2, the interaction is specific. You can't just take any old first protein. It has to be the actual one that matches within the operon. It's specific interaction. That means the amino acids are particularly programmed to be like a lock and a key. Like a lock and a key. Okay? Now this was important because, so we previously found the protein. We found that it was an operon. We postulated that it was a toxin-antidote operon, but now we've actually proven that it has behavior that matches a toxin-antidote operon, right? Does that make sense? So that was a big deal. Okay, next figure. Next figure is, how much time do I have? I got plenty of time. Okay, so this figure is testing the toxin-antidote hypothesis in yeast, okay? So we've learned from yeast how plasmids work. You can use a GAL1 promoter. Did I talk about what turns that one on? Galactose. And what shuts it off? Glucose shuts it off. So here is a negative control right here. In these lanes, none of these proteins are turned on because they're in the presence of glucose. So this is off. In this lane, are the proteins on or off? They're on, because this is all in the galactose system, okay? And I can get different plasmids. And on one of these plasmids, I put a Ura3 cassette. What does that mean? Right, that's the selection in the yeast. And in one of these plasmids, I had a Lu cassette. What's that? Leucine, that's the other selection. And in one of these, I made it a 2 micron. What's that mean? Copy number. Is it high or low? It's high. So this is about 60 copies. And in one, I made it a sen. What's that one? That's right. It's 1 to 3 copies. So it's low copies. And on the low copy plasmid, if I wanted to set up a system where I can test what happens to the toxin in the presence of the antidote. So I need 2 plasmids, okay? And the reason I changed the origins is because I can't have 2 of the same origin, or they'll compete. So I needed to set up a system where I could test 2 different plasmids in the same cell. So they need different selection cassettes. One needs to be in different origin. One a low copy, one a high copy. I put my toxin, SID-B, in the low copy, okay? Rationalizing that if it's toxic, should just need a little bit of it. And the antidote, you might need a lot of it, right? Because remember from that mating experiment, it's being stockpiled. You might need a lot of it. So that's why I put the antidote in the high copy 2 micron. Okay. So that's this data. So what's LAC-C again? It's a negative control. Nothing should happen if you overexpress LAC-C in yeast. It should be fine. So what you're looking at here is a serial dilution. This is just measuring the growth of yeast. This is very, very common in yeast biology. You'll see this, and what they're just measuring is there any effect on the growth of the yeast from overexpressing these proteins? So there's no effect on the growth of the yeast, and the way you can conclude that is because this galactose looks exactly the same as the glucose. So when the protein LAC-C is being turned on, it looks exactly the same as when the protein's off. So there's no effect on the yeast, okay? SID-A, that's the antidote, okay? No effect on the yeast. If you overexpress the toxin, what happens here? It dies, it kills the yeast. And then what we did is we made one construct, back to my notes, we made one construct where I made a mutation with a quick change, and I mutated C1025A. What's that mean? Cysteine to an alanine. And we knew that this SID-B toxin was likely to be an enzyme because we could see this catalytic cysteine at a special spot. So we thought it was a particular type of protease. And to validate that it was a protease, we took the active site, the active amino acid, which catalyzes the reaction, and we mutated that cysteine to an alanine. So that's the SID-B star. So let's look at the data. What happens in the SID-B star? They live. So that proves that the toxic activity of the toxin is due to that protease domain, that enzymatic domain. So we figured out what it was, essentially, how it was doing what it does. Okay, this is the paralog version. So SID-B is a deubiquitylating enzyme. Talk more about that later. SYN-B is a nuclease. So here we did the same thing. The antidote for SYN-A, no effect. Toxin for SYN-B, it kills the yeast. And we found the catalytic residues in the nuclease, which are a D, an E, and a K. They hold the magnesium ion. I've talked about this. And that's one reason why you add ADTA to lysates to chelate these magnesium ions. So we mutated the DEK to alanine, alanine, alanine. That's the SYN-B star, and it grows. Okay, so that means that the nuclease activity in the nuclease toxin is due to that nuclease domain. Okay, so we validated that these 2 toxins are actually toxins, and they both behave, they bind each other like toxin-antidote systems. So we got binding, we got specific binding, we got toxic activity in the yeast. Now we needed to test what happens if you put the toxin in the presence of the antidote. So this plus this in yeast. So we did that here. Okay, so here we're mixing and matching the 2 different antidotes with the 2 different toxins. So here we have SID-B. Here's a negative control. SID-B just plus an empty vector, and it's dead. Here's SID-B plus the wrong antidote, SYN-A, and it's dead. Here's SID-B plus its matching partner, and what is it? It's alive. So it's a rescue. It's a rescue of the antidote with the toxin. Here we have the second thing, the second paralog. SYN-B, the nuclease toxin, with an empty vector, it's dead. SYN-B plus its matching partner, it's alive, it's rescue. SYN-B plus the wrong antidote, and it's dead. And then here's the positive, here's the negative control where all the genes are off and all the yeast grow fine. So we then proved that if you mix and match, you also, not only do you get the specific binding, but that specific binding correlates with specific rescue in yeast. So we basically validated a rescue system in yeast. Okay. So then we did all that work. I might come back to this. This is us validating what type of enzyme it was. Here's more of the biotechnology stuff rather than the biochemistry. So we validated that in yeast, this thing behaves as a toxin-antidote system. The final question was, if this is the hypothesis or the question, if this is the CI system, can we make synthetic CI in an insect without Wolbachia? So can we put the CI genes in any insect and cause sterility? Why would we care about that? Why would we want to do that?
Or mosquitoes.
Yeah, you could do the sterile insect technique. Or maybe you could do gene drive. If you put in both the genes, like maybe you could make it work. Maybe you could get sterile insect technique or gene drive without Wolbachia. And maybe then you're controlling for certain things because you don't actually have to use the bacteria. Okay, so there's some subtle advantages to that. It makes the system a little bit better. So the question was, can we insert these genes in an insect through a transgenic system and induce sterility? So what we did, here's our fly vector. Okay, so UAS, that means it's a GAL4 UAS system. That means we can cross it to a driver if we want, and that driver should turn the genes on. It's got a promoter. It's got a P element promoter. What's the P element? It's a transposon. So this is a promoter for a transposon. Where's the P element active? Does anybody know?
The ATTB side?
No, it's active in the germ. Well, that's a landing site. It's something different. The P element transposase, I mean, like when does this promoter for the actual gene turn on? It turns on in the germ lines. So this is kind of like a germline promoter. We had a UTR, a K10 UTR. I don't know if that's worth talking about. Here we have, what's this? Remember? This is the thing that recombines with the landing site, the ATTP site on the chromosome. So this is a Phi C31 integrase insertion vector that's going to do a site-specific insertion. What's this? Right, so this is going to cause the fly eyes to turn red. This is the origin for E. coli, so you can grow it in E. coli. This is the selective cassette for E. coli. So then we took our operon, we took the whole gene, and we popped these different things into the multi-cloning site of this plasmid. What's this?
T2A.
Yeah, well, I've talked about that before. What is that?
It's like a peptide. Like you cut it right there or something? Or
Yeah.
it skips?
Yeah, it skips. So we wanted to express both the toxin and the antidote. So if this is the toxin, this is the antidote. We wanted to see what happens if you express both at the same time, just kind of like a normal system. And we wanted it to behave kind of like an operon, but eukaryotes, flies, don't have operons. So we needed to program the system to just have one insertion of this large ORF and to translate two proteins, this one and this one. Okay, so we made those insertions. We verified that they are being expressed with RT-PCR. And then here was our big final concluding figure. So here's what happens when you have the gene, when you have the operon. This is progeny. So this is, we're just mating these and we're counting how many offspring they produce. What happens if you have the operon in the male? You're dead. They're all sterile. All the males are sterile. What happens? Here's operon star. Here's the catalytic inactive. It's fine. This proves that the sterility is due to that enzymatic deubiquitylase domain. Here's the reciprocal cross. So here's a wild type male crossed to a female with the operon. What does this tell you? No.
That it's the males carry the toxin.
Right, it's sperm specific. It's not doing anything to the eggs of the mom. Does that make sense? If you have it in the mom, it doesn't do anything to the eggs, which is consistent with the bacteria. The bacteria don't do anything to eggs. They just mess up the sperm. It's only messing up the sperm. You only get the sterility phenotype when it's in a male. Okay? And here we're just showing, these are negative controls to show you that if you overexpress any old gene, here's the antidote or the GFP. If you overexpress any old gene in the male, you don't get the sterility. So it's specific, and it's because of this dub domain. So we proved that we could put in a gene that would sterilize an insect in fruit flies with the CI system. Let's see. I could talk about this, but let's talk about some of the problems we had, because this is good, like brainstorming. Okay, so we tried the experiment. What's the rescue experiment? We tried the rescue experiment. What is it? How would you do it?
In yeast.
No, in flies. We proved it in yeast, but like I didn't show you a figure of rescue in flies. What would the experiment be? It would be a cross, right? And what would be the genotypes? Well, this is synthetic, so not Wolbachia, but the operon. So this would have the operon, and this would have either the operon or the rescue factor, SID-A, right? I tried these experiments. They're all still dead. What does that make you think? What's your conclusion from that?
Maybe the females they aren't producing you.
Yes. Yes. So our paper, we sent it to Nature, and they rejected our paper because we said that and they didn't believe us. They said that and they didn't believe us, so they rejected our paper at Nature, and then they sent it to Nature Microbiology. Those fuckers. Oh, I'm so mad. But yes, that was our rationale. Was we thought we just couldn't get enough expression. We couldn't get enough of the A that was needed to get the rescue. And then what happened later, somebody changed to a different driver. So there's different drivers. You can like enhance the expression. Somebody did an experiment where they took maternal triple driver, which is a big gal4 driver. And then they did SID-A overexpression. And then they took a W-mel infected male. So wild type CI. And they did this cross, and they were able to get rescue. Just as a quick update, we did get the rescue experiments finally in the Beckmann eLife 2019. We tried all that stuff, but we got it to work in the yeast at least. Like we felt like that was enough. Plus we had all the biochemistry. We knew exactly what type of enzyme it was. We knew how it worked. The other people did not have any of that stuff. So I felt like my paper was better than the other paper. And I'm actually on the other paper as well. But anyway, that's that. So that's CI. So now this is important. Like, why do we care? I wanted to go through all the data because it's kind of a good summary of all the techniques that I've been teaching you guys, like all the yeast stuff, the promoters, all that stuff. Like that's what it takes to get a whole synthesis of a good project in biotechnology. And why is it useful for biotechnology? Well, now imagine we know the CI system. So imagine you have a problem insect that you can't irradiate, and you can't get it infected with Wolbachia. You could maybe just insert the transgenes of the CI system into the insect, have it cause the sterility, and then use it for the sterile insect technique. So it's kind of like a platform technology from which we could do any sterile insect technique. And that's where I'll end it. That's my story.
So how exactly does the SID-B stop the sperm from being viable?
That's what my next paper is. It's in final drafts right now. I don't have enough time to talk about that, but I'll say briefly that it's a deubiquitylase, so that means it removes a post-translational modification from some substrate. And we've been trying to figure out what's the substrate that it's targeting. And there's a process in sperm that when they become adult sperm, they need to protect their DNA because it's a germline, right? They really care about their DNA. They don't want any mutations. So they remove histones and they put in these proteins called protamides, which basically like compact and protect the sperm. And there's a defect in that process that we think is caused by the deubiquitylation of a key factor that grabs the protamines and takes them out and then replaces them with histones. So it's in the process of the protamine histone exchange. That's kind of might be a little bit too detailed, and I don't have enough time to like explain it all, but that's what we think. That's the next paper. That should be out within a year, hopefully. And that's out now. Beckmann eLife 2019.